Biology 9700, paper 11, May, June 2024, question number 1 to question number 20. So first of all, we will move towards the question number 1, and you can see question number 1. The diagram shows a stage micrometer scale viewed with an eyepiece reticule using a magnification of 200. Here you can see the micrometer scale. And the lower one is the eyepiece reticule. So we have to calibrate these two values of micrometer and eyepiece reticule. So it is eyepiece reticule value is 0.1 millimeter. We have to convert this 0.1 millimeter into micrometer. So we will multiply 0.1 with the 1000. So the answer will be equal to 100. So 100 micrometer is equal to 40 units of eyepiece reticule. So we will divide this 100 with 40, which will be equal to 2.5. So we will multiply this 2.5 with 4. So the answer will be 10. Or 10, we can write 10 micrometer. We can write in this way 1.0 into 10 raised to power 1 micrometer, and it is equal to the A. So A is the correct option. Now we will move towards the question number 1, from question number 1 to question number 2. So the question number 2 is, which range of all diameter is typical for prokaryotic cells? And prokaryotic cells have their, you can say, size, which is 1 to 5 micrometer. So A is the correct option in this case. Now we will move towards the question number 3. And question number 3, you can see the diagram shows a typical animal cell, and they have labeled 3 different parts. Where would nucleic acid be found? So nucleic acid, we found the part is cytoplasm, and second part is ribosomes on endoplasmic reticulum. And then third is the mitochondria. So all of they have the nucleic acid in them. As you know, the nucleic acid is present in the form of chromosomes, and in case of protein synthesis, and then this messenger RNA comes out of this nucleus and goes into the cytoplasm on the ribosomes, and their translation takes place. And in this way, mitochondria provides the energy, and mitochondria also has nucleic acid inside. So I would like to say the A is the correct option. So we will move towards the question number 4. Which structure is found in a typical bacterial cell? Intron, telomers, template strand, and capsid. So intron, so this is not the correct option. Telomers, you know, they are present at the base of chromosomes. You can say which save the genetic information. So template strand, it can be the correct option, and capsid also for the protection give and present in the virus. So C is the correct option in this case. What is the maximum number of hydrogen bonds that can form between a single water molecule and other water molecules? And here you can see the water molecule is made up of oxygen and 2 hydrogen ions, which they have partially positive and negative charge on them. So due to this partially positive and negative charge, you can see that they can make bond with other molecules. So in this case, they can make, because in the last, in the, there are 1, 2, 3, 4, 5, 6. 6 electrons are there in the shell. So if two of them are, you can say, combined with the other water molecules, so in this way, 1, 2, so they can make the 4 bonds with the other molecules. So in this way, D is the correct option. Question number 6. And question number 6 is, the diagram show the structure of 4 amino acids in aqueous solution: glycine, lysine, alanine, and aspartate. Which two substrates have an overall charge? Overall charge, this is the minus and plus charge, so they will cancel the effect of each other. So this is not the correct option. And alanine, they will also single effect, the cancel the effect of each other. But here on this lysine, you can see minus charge, positive charge, and positive charge. So there will be the overall charge, that is the plus charge will be more than negative charge on the lysine, can be the part of the answer. So aspartate, you can see negative charge, positive charge, and there will also be one more negative charge. So in this way, it can be the part of uh, the answer. So A, alanine and aspartate, no, it is not the correct option. And alanine and glycine, no. Aspartate and lysine, C. And glycine and lysine, no. So C is the correct option of the question number 6. We will move towards the question number 7. And question number 7, the table shows the number of carbon atoms and the number of carbon to carbon double bonds in molecules of 3 triglycerides and the fatty acids they contain. The information is shown in the format number of the carbon atoms, number of carbon to carbon double bonds. So which description matches fatty acid 2 of the 56:4 triglyceride identified in the table at X. So here you can see that uh, fatty acid, one fatty acid will always combine with the 3 uh, there will be the 3 carbon and there will be the 3 fatty acid chains with them. So in this way, there will be the formation of 1 glycerol and 3 fatty acids. They will make up triglycerides. So triglyceride will join with 3 fatty acids, fatty acid 1. So 15, 18, and 18, if we count them, so it will not be equal to 54. So this is, and the other 17 and 18. And if we add up them, so it will be equal to 18 carbon atoms. So 57:3 ratio, which description matches fatty acid 2 of the 56:4 triglyceride identified in the table as X. So saturated with 17 carbon atoms, no, it will not be the correct option. Saturated with 18 carbon atom, again, it will not be the correct because it will be the unsaturated. So both of they are unsaturated, C and D, but 17 carbon atoms, so no, it is not the correct option. So unsaturated with 18 carbon atoms, so it will be the correct option. D is the correct option in this case. So here will be the answer 18. So we will move towards the next question. Question number 8. And question number 8 is, collagen molecules are made up of 3 polypeptide chains interacting together. The individual polypeptide chain consists of a regular pattern of amino acids. Almost every third amino acid is glycine. Which protein structure of the collagen are described? It means the polypeptide chain interacting together. The individual chain consists of a regular pattern of amino acids. Almost every third amino acid is glycine. So it can be the primary structure. And made up of three polypeptide chains interacting together. Three polypeptide chains. So it can be the quaternary structure. So primary, and this is the combination of two chains, that is primary and quaternary. So B is the correct option. The masses of the parts of the hemoglobin are shown in the table, and you can see there are a table is given components alpha globin chain and beta globin chain, and then heme group, and they have given the mass in the other column. There are 1000 Da in 1 kilo Da. What is the mass of a hemoglobin molecule in kDa? So here you can see that we will multiply it with 2, and it will also be multiplied with 2, because there are 2 alpha globin chains and there are 2 beta globin chains, and there are 4 heme groups because there are 4 chains. So there will be 4 heme groups. So by multiplying them, we can get the answer, which is D. So D is the correct option for of the question number 9. So we will move towards the next question, and next question number 10. What is the correct order of location in the cell for the production of extracellular enzymes? In extracellular enzymes, you know that their protein enzymes are protein in nature. So what will happen? What will be the location? You can say correct order of the location. So first of all, it will start in the nucleus, and from nucleus the messenger RNA will take the messages towards the ribosome, and then from the ribosome there will be the formation of proteins and the enzyme, and then it will be stored in the endoplasmic reticulum, and then from endoplasmic reticulum modification will take place in the Golgi body, and then it can you can say secreted out of the cell. So it will move out of the body. So A is the correct order. So B, C, and D are not the correct options. Only A is the correct option in this case. Now we will move question number 11. So question number 11 is, the graph shows the trend from an enzyme catalyst reactions. Which labels are correct for the x-axis and y-axis? And x-axis and y-axis, you can say they have given us 4 options. Rate of reaction. So rate of reaction is always, because this is dependent value, so we will take it on the left side, rate of reaction. And substrate concentration, enzyme concentration, temperature, pH, rate of reaction, substrate concentration, all other, that is substrate concentration, enzyme concentration, pH, and substrate concentration and temperature, all they are the independent values. So the independent values are always taken on this x-axis. So here you can see the rate of reaction is different at every moment, but it is maximum at the center. It is maximum in the center. It shows the values, as you can see. It shows the value different. It shows the different values. So the values which can be present in a pH, because if we take the temperature, so temperature, there will be the graph just like this. The graph will be just like this. So there will be at 40 degree, but it is in the center. So it is in the center. It shows the pH. So optimum pH, which is 7. So it is in the center. So the answer can be the, on y-axis there is rate of reaction, and on x-axis it is pH. So C is the correct option. A, B, and D are not the correct options. We will move towards the next question, which is question number 12. And question number 12 is, which statements about the Michael-Mention constant are correct? The higher the Michael-Mention constant, the enzyme's affinity for the substrate. So I think this is not the correct option. So higher the enzyme affinity, no, it is not. It is inversely proportional. Chem is a measure of the degree of enzyme affinity for the substrate. It is correct. Is defined as the substrate concentration and which the enzyme functions at half its maximum rate. It is also correct about Michael-Mention constant. So according to this, these statements, the D is the correct option. A, B, and C are not the correct options. Next question is 13, question number 13. The diagram shows how nicotine is transported from the blood plasma into a cell using a type of uh, a co-transporter mechanism. So according to this blood plasma has been shown the cytoplasm of the cell. And here you can see cell surface membrane is there. So co-transporter protein is there, which will take the protons, move down their concentration gradient out of the cell. So protons or hydrogen ions, they will move against the, you can say down their concentration gradient. It means from higher concentration towards the lower concentration. At they in the cytoplasm, they were in greater concentration. So they moved out into the blood plasma to the co-transporter proteins. And on the other hand, the nicotine is transported against the concentration gradient. So here the nicotine was in lower concentration, and from its lower concentration, it moved towards its higher concentration, that is transported against its concentration gradient into the cell. So in this way, in the phloem tissue, there is a co-transporter mechanism that moves the sucrose into the cytoplasm of a companion cell. Which statement correctly describes us similarly between the co-transporter of nicotine and the co-transport of sucrose? Yes, we have, for this purpose, we have, first of all, we have to draw this phloem cell, and there is a companion cell with it. And here you can see the food which is manufactured in the phloem, and from the phloem in the mesophyll tissues, food is manufactured over there, then it moves towards the phloem, and phloem take it to the different parts of the body. So what happens over here when a companion cell has nucleus inside? So it helps the this transport of sucrose towards the different parts. So here what happens? At that time, there are co-transporter proteins and co-transport mechanism in the companion cell. So what will happen? First of all, the hydrogen ions, they will move down their concentration gradient out of the cell in this way. And but at the same time, there will the sucrose has to enter, and when sucrose has to enter into them, so at the same time, these hydrogen ions will also enter into this. So both of they will enter against their concentration gradient. So they will move against their concentration gradient, but with the help of facilitated diffusion. So difference is there, that is hydrogen ions when they move out and nicotine move inside the cytoplasm. But here this case when hydrogen ions or the protons move out of the, but they re-enter again with the facilitated diffusion. So we will move towards the statements. The co-transporter proteins generate a proton gradient by moving protons out of the cell by the active transport. No, they will not move by the active transport. So they will move from their higher concentration towards the lower concentration. The protons are transported through the co-transporter protein by facilitated diffusion, so it can be the correct option. The protons move through the co-transporter proteins in the opposite directions to the movement of the nicotine. No, they will move in the same direction over here. Uh, so it is different in both of these cases. The co-transporter proteins use energy from ATP, so they don't use energy. They move with the help of facilitated diffusion, so they don't need energy. So it is also the incorrect option. Only B is the correct option in question number 13. So we will move towards the next question number 14. And question number 14, you can see the diagram shows a simple metabolic pathway. Simple metabolic pathway W to X to Y and Z. The letters W, X, Y, and Z represent four different substances. So they are four different substances. At each step in the diagram, the substrate undergoes a chemical reaction catalyzed by an enzyme. So one is changed into other, and W is changing into X, X into Y, and Z. All they need the different enzymes to change. The reaction produces the next substance in the pathway. Which statements correctly describe the enzyme taking part in this metabolic pathway? They all are globular protein. Yes, all are globular protein. So this is the correct statement. And then they all have the same tertiary structures? No, they will not have the same tertiary structure. They have different active sites according to their substrate. So this is not the correct statement. They all contain hydrogen atoms in their structure. So this is the correct statement. So according to these two correct statements, statement number 1 and statement number 3 is the, so C is the correct option. Now we will move towards the next question, question number 15. And question number 15 is, a student observed the effect of the different concentrations of the salt solution on blood cells. The student added each concentration of salt solution to one of the two microscope slides, and then a small drop of fresh blood was added. Each slide was viewed using the high power lens of a microscope, and the students' observations were recorded. Slide 1. The red blood cells were, no red blood cells were visible. It means all of they were busted. So there was no red blood cells over there because the water has entered into them, and due to entering of the water, all they got busted. The red blood cells were visible but looked slightly crinkled. It means they were kept in the solution of less water potential. So the water moved out of the red blood cells, due to which they will shrink or they were crinkled. Which row contains the results obtained? Slide 1, swelling of the cells caused them all to burst. Yes, this statement for about slide 1 can be correct. The water potential of the cell was more negative than the water potential of the external solution. So no, it was opposite to this statement. And B, the water potential of the cell was more negative than the water potential of the external solution. So it is the correct statement. So due to this reason, the water was entered into the cells, due to which they were busted. They were not visible because they were busted. The water potential of the external solution was more negative than the uh, water potential of the cell. That is the reason the water was moved out of the cells and it was plasmolized. The water potential of the cell was less negative than the water potential of the external solution. So there is a net movement of water out of the cell by osmosis. So the water potential of the external solution was less. The cell, the more less negative. Again, this is not correct. So in this way, I would like to say B is the correct option. Question number 16. So question number 16 is, which statement correctly describes facilitated diffusion? The process only occurs using channel proteins that change shape and that use energy provided by the cell. They don't change the shape. So this is not the correct option. The process occurs using channel proteins or carrier proteins that may or may not change shape. It can be a correct option. The process occurs using channel protein or that use energy provided by the cell. Facilitated diffusion do not need energy. The process only occurs using carrier proteins that create a gradient to more ions in the opposite direction. Not a correct statement. Only B is the correct option. Question number 17. And question number 17 is, which row is correct for stem cells? Can repair cells? No, they cannot repair cells. They can repair the tissues, but not the. So can be involved in the formation of phagocytes. So they are involved in the formation of phagocytes. So according to this, D is the correct option. A, B, C are not correct. Only the D is the correct option. Question number 18. So question number 18 is, eukaryotic organisms grow and increase in size as a result of cell division. The diagram shows some stages of mitosis with one stage missing labeled X. What happens to stage X? Spindle fibers attached to the centromere and the chromosomes line up along the equator. No, they are lined up upon, this is the metaphase. The chromatin coils and condenses and the nuclear membrane disintegrates. The chromosomes line up in pairs along the equator of the cell. No, no. Molecules shorten so that spindle fibers contract and the centromere divide. D is the correct option. As you know, student, this is the metaphase. And this is telophase. And microtubules shorten. As you know, the chromosomes has to move towards the opposite poles. So in this way, they are the microtubules are get shorter. So D is the correct option. Now we will move towards the question number 19, and question number 19 is, here you can see the graph shows various distance measurement taken from the start of metaphase of mitosis. The graph are to scale when compared to one another. So the distance, time and distance. Here you can see, first of all, there is an equal distance and distance is decreasing. So distance is decreasing, but in this case, you can see the distance is increasing, then it is becoming equal. So we are moving towards the distance between poles of the spindle. So this is not the correct option, because between poles, the distance is not decreasing. The distance between poles and the distance spindles, it is not decreased. Firstly, then distance of centromere from poles of the spindle. No, this is not the correct option. Distance between poles of spindle. No. And then the distance of centromere from poles of the spindle. From poles of the spindle, again, it is not the distance of centromere from poles of spindle. Distance of centromere from poles of the spindles. So these are the poles, and in the metaphase, they are, and then they start moving, and there will be the. So this is the correct distance between sister chromatids. So distant sister chromatids, the distance when they move towards the opposite pole, the distance between is increased, and the distance between the pole is decreased, and distance between sister chromatid is increased. So distance of centromere from uh, poles of the spindle, and the distance between poles of the spindle. No, centromere again. C is the correct option. As you know, what will happen when they move towards the opposite poles? So the distance of the centromere from poles of the spindle is decreased. You can say this is decreasing, and then the distance between sister chromatids, because they are getting separate from each other. When they divide at centromere, they are getting separate from each other, and the distance between them is increasing. So the C is the correct option in this case. Question number 20. So question number 20 is, question number 20, little bit you can see. We are moving towards the question number 20. Which row is correct for the start of anaphase of the mitosis? So form of DNA chromosomes, anaphase, start of anaphase mitosis, separated sister chromatids. So these two can be the option C and D. So the DNA is associated with stored protein, correct? State of cell surface membrane. State of the cell surface membrane can be broken apart, intact, intact, broken apart. So it will be intact, always intact. So C is the correct option. It will not broken apart. So this is not the correct. Sometimes again, it is always. So student, it was almost from question number 1 to question number 20. Please like, share and subscribe my channel to watch more videos. Thanks for watching.